Physiology of Gustation or taste, physiology of taste. So let us see some of the general points. Like taste buds contain taste receptor cell. The papillae, that is taste buds, that is the papillae are fungiform, which is distributed throughout the dorsal surface, about 2/3 of it, of our tongue. Then velate, caudal portion of the tongue, and foliate, dorsolateral part of the tongue. So dogs seem to have sense of taste for sweet, salt, sour, and bitter. However, they do not have highly sensitive salt receptor. Dogs have about 1,700 taste buds, while humans have about 9,000, much more than the dog. So humans are more able to discriminate taste than dog or animals. Dogs have considerably more taste buds than cats. Cats have only 450, around 450 taste buds. And one thing we have to remember that olfaction and gustation are both intermingled. So it says that taste is 80% of smell. So taste is 80% of smell. So it is both are very much intermingled. Olfaction and gustation. So taste buds. The basic flavor are meat, salt, sweet, sour, bitter. Taste buds are composed of group of about between 50 to 150 columnar taste receptor cells, which is bundled together like a cluster of bananas. So this is an example of a taste buds which have many taste cells. So within the taste bud, and each taste cell have receptors for only one type of flavor. One cell sends only one flavor. Thus, each cell in a taste bud detect either sweet, sour, or bitter, or salt. Let us see how the taste discrimination or the taste cell receptors are stimulated. When the molecule, it may be salt, it may be hydrogen ion because of sour or bitter chemical, or sweet like polysaccharide or glucose. When this molecule, first they have to dissolve in the saliva. After dissolving in the saliva, this molecule they enter into the taste bud through a pore. So this is a taste bud, this is the pore, and through the pore, those molecules enter and they bind with the microvilli of the taste cell, and the taste cells are depolarized. So they enter into the taste bud through a pore, bind to the receptor, which is present in the membrane of the microvilli. And after binding of the receptor molecule to their receptor, the cell is depolarized, taste cell. The taste cell that may be salt or sour or sweet, bitter, umami or meaty or savory taste. So when the salt, how the salt stimulate the depolarization of the taste cell? First, when the salt, they will stimulate the amyloride sensitive sodium channel, and because of the sodium enter into the cell, and this entry of sodium stimulate takes the cell to its threshold potential when the voltage gated calcium channels are stimulated and the calcium entered inside. And when the calcium entered inside the cell, the vesicles within the cell which contain the neurotransmitter, those vesicles are fused with the cell membrane and release the neurotransmitter by exocytosis. So that neurotransmitter may be ATP or serotonin. So the hydrogen ion, this hydrogen ion prevent the potassium leave from the cell, thus cause depolarization. We know that potassium, the cells have the leaky potassium channel. So when the potassium is prevented by the hydrogen ion due to the this hydrogen ion, when it prevents the potassium to leave from the cell, then the cell, the positive, the negative, the cell resting membrane potential of the cell, it takes to its threshold potential, as well as soon as it reaches to its threshold potential, the voltage gated calcium channel opens and the calcium enter into the cell, and the synaptic vesicle, they release the neurotransmitter and stimulate the sensory neuron. So sour substances, they have the hydrogen ion. Because of hydrogen ion, they cause stimulation of the this receptor cell, these cells which are sensitive to sour or hydrogen ion. And sweet, bitter, umami, meaty or savory, their binding site almost is the same. They bind with the G protein couple receptor, and after binding with this molecule like glucose, like polysaccharide, or bitter like caffeine or quinine, other substances, when they bind with the G protein couple receptor of the receptor cell, then it stimulates the phospholipase C, and ultimately they form the IP3. This is the second messenger. This IP3 directly stimulates the calcium release from the endoplasmic reticulum. When calcium comes from this reticulum, they cause synaptic vesicle to fuse and release neurotransmitter and stimulate the sensory neuron. So this is, we have the taste bud, and this is this cell, this receptor cell sensitive for salt. This salt, it stimulate the, or this amyloride sensitive sodium channels, through it the sodium enter into the cell and cause stimulation of voltage gated calcium channel or calcium channel, and calcium come inside and cause synaptic vesicle release the neurotransmitter by exocytosis, and this neuronal end are stimulated, sensory neuron. So sweet, they bind with the G protein couple receptor and they activate the phospholipase C. This phospholipase C break down the IP2 from the membrane into IP3 and DAG. IP3, they directly stimulate the calcium release from endoplasmic reticulum, and they cause the release of neurotransmitter and stimulate the sensory neuron. Okay. So when the cell is depolarized, taste cell, the taste cell may be sensitive for salt or sour or sweet, bitter, meat. So we have talked about their pathway, how the cells are, the receptor cells are stimulated. When receptor cells are stimulated, they release the neurotransmitter, and the neurotransmitter bind with the sensory neuron, dendrites of the sensory neuron, and the sensory neuron generate the axon potential. These sensory neurons, they carries the information to the medulla, that is the nucleus tractus solitarius. So here the sensory neuron from the tongue, like facial nerve, cranial nerve 7, and glossopharyngeal nerve, cranial nerve 9. And also one more vagus nerve, cranial nerve 10, that carries the taste buds present in the epiglottis or the pharynx. From there, the vagus nerve also takes information to the tractus, nucleus tractus solitarius. I will show the diagram. So here I have shown the in dog, or I have shown here two cranial nerve 7 and cranial nerve 9. That takes the information to the nucleus tractus solitarius. From there it goes to the hypothalamus, then to the insula, the cortex, where the gustation or the taste discrimination occurs. And these fibers also send information to the other parts of the cerebral cortex like hippocampus, hypothalamus, and amygdala. So these are the taste cell. From there, the sensory neurons are stimulated, and through the dorsal ganglia, it enters into tractus solitarius, the glossopharyngeal nerve, and facial nerve also through geniculate ganglion enters into or synapse into tractus solitarius. From there, it goes to the hypothalamus and to the center where the taste discrimination occurs. So this is the pathway how we sense the taste. So let us self-evaluate whatever we have learned just now. Okay, taste molecule binds to receptors on the microvilli of the receptor cell. Because microvilli of the receptor, the cells have they bind with the like G protein coupled receptor or the sodium entry amiloride sensitive sodium channel opening. So cranial nerve that mediates signal for taste and facial and glossopharyngeal nerve. It is true. Which statement is correct about detection of salt test? Ligand bind to G protein coupled receptor, amiloride sensitive sodium channel trigger, influx of sodium, depolarizing cell. ATP is the most likely transmitter that depolarize sensory terminals, and phospholipase 3 involved in signaling transaction. So salt taste, it is first, this is the correct answer is the B. Amiloride sensitive sodium channel trigger the sodium influx and cause depolarization of the receptor cell. The primary afferent fiber that mediates taste signal terminates in the nucleus tractus solitarius. So in the next video, I'll be discussing a little bit about the amiloride, a sensitive sodium channel. So this is about the physiology of taste or gustation. Okay, thank you.